Can COVID is an open science collective dedicated to rapidly mobilizing and sharing knowledge to help inform Canada's COVID-19 response. To learn more about us or how to join our community, you can visit our website at cancovid.ca.
So I think we're ready to get started. Hello, everyone, and welcome to this week's edition of Can COVID Speaker Series. Thank you for joining us today. My name is Rosa, and I'm the Knowledge Translation Manager for Can COVID and also the moderator of the Speaker Series sessions. And it's my pleasure to introduce to you to this week's speakers, Dr. Parkins and Dr. Pang. Dr. Parkins is an associate professor at the Department of Medicine and Microbiology, Immunology and Infectious Diseases at the University of Calgary, and also a section chief of Infectious Diseases, Calgary Zone, Alberta Health Sciences. And Dr. Pang is a molecular virologist at the Public Health Laboratory of Alberta, Precision Laboratory, also a professor at the Department of Laboratory Medicine and Pathology at the University of Alberta. And today's title is COVID-19 Data Modeling from Alberta's Integrated Wastewater Monitoring Program. And so without further ado, I will hand over the floor and the mic to Dr. Parkins for the presentation today. Thanks all.
Thanks very much for having us. I will initiate the presentation and halfway through turn it over to my co-host, Dr. Xiaoli Pang. We do not have any conflicts related to this work that we need to declare. What we're here to talk about is wastewater-based epidemiology, and this is an emerging area of science that seeks to understand a population by analyzing various chemicals in wastewater from the community with which it derives. And this is very much similar to looking at blood samples from a human host to understand the health of the human host. The difference being that with wastewater, by going at a more and more granular view within a sewer shed, we can get more and more precise information to different parts of the organism that we're monitoring, in this case, a community. What's important about this technology is it's comprehensive, capturing everybody contributing to the wastewater network. It's inclusive and doesn't have any biases that exclude marginalized groups. And it's, of course, objective and unbiased. And it's possible to use wastewater-based epidemiology to understand community COVID-19 disease burdens because SARS-CoV-2 RNA is excreted in the feces of infected individuals. And because it's excreted in the feces of infected individuals, we've been able to detect it in wastewater. And teams world over have been able to demonstrate that wastewater-based epidemiology is a very effective mechanism by which to do COVID-19 data modeling in communities. And again, what's so important about this technology is that wastewater-based epidemiology captures those with symptoms and those without, and those that seek to get testing and those that either do not or cannot. So again, it's an inclusive technology. I'll very quickly walk you through our workflow from sample collection, cleanup and concentration to remove those impurities, the molecular analyses we do that are very similar to what clinical laboratories do to make confirmed diagnoses. We'll focus my talk on how we present this data, and we won't specifically go into detail on how this data is used by our partners, whether it's the medical officers of health and Alberta Health Services, or the emergency operation commands of each of the municipalities of Alberta that are involved, including the largest, Calgary and Edmonton. So we collect wastewater at least 3 times a week from a range of facilities across Alberta, whether it's the large wastewater treatment plants or individual facilities or sub-sewer shed connections in neighborhoods. And we're going to focus this talk specifically on the wastewater treatment plants because, again, these are the largest means by which to monitor an individual community. What our wastewater samples are 24-hour composite samples, allowing for us to understand the time variations in wastewater flow received at each wastewater treatment plant, which purport to the actions of humans in the toileting patterns. Our wastewater monitoring program looks at 22 different wastewater treatment plants monitoring 29 different communities across Alberta. We've specifically targeted all large municipalities greater than 8,000 individuals, focused on specific important trade and commerce hubs in the province, looked to achieve good geographical representation, and our goal has been to provide real-time monitoring of at least 75% of Alberta's populations, and we are exceeding this already. Our objectives of this are to rapidly collect samples, process them within 24 to 48 hours to allow for real-time leading indicators of wastewater to suggest what the COVID-19 community burden is going to be. The main goal of our program is data sharing with knowledge users and decision makers at all levels, but also transparently sharing the data with the communities and the general public. There's two teams, and over the course of this last 2 years, we've developed different mechanisms by which to process our wastewater. They are very similar. What we do is collect wastewater from each site. We then spike that wastewater with an internal control that we can then assess for to make sure that there's no inhibitors present in our final sample. The dilute matrix of wastewater is concentrated, and we remove impurities and factors that which would result in RNA signal degradation. We then use molecular analysis to target specific components within the SARS-CoV-2 genome, either the nucleocapsid gene regions N1 or N2, or the envelope gene, which we've used in the past, our spiked control, and intrinsic markers present in feces that can indicate the amount of fecal matter present. We use, of course, the same clinical tools labs use, RT-qPCR, to quantify how much of each target is present in each wastewater sample. And what I propose to talk about today is how we are reporting this, which is through the University of Calgary's Cummings School of Medicine Center for Health Informatics. And partnering with this group was obvious because they maintain an interactive COVID-19 data sharing platform and have for the first 2 years of the pandemic. In the first year, we had a Calgary-specific wastewater monitoring program, and this last many months, we have a wastewater monitoring program that serves all of Alberta. And I'll take you to that website right now. So here is the website for the Center for Health Informatics, and we'll go to the wastewater tab. And here are the communities that we're currently monitoring. So right now we're demonstrating Calgary, and what you can see is that we've plotted individual data points and then have the rolling average of those data points to indicate the trends in which SARS-CoV-2 RNA values are trending in communities over time. We display the N1 and N2 regions and the average. And we look at, again, either the rolling average to focus on those trends, which are so very important, as opposed to any individual data point, because wastewater is a complex and heterogeneous matrix, or you can look at the default as exact values as well. We're monitoring major programs in cities across the province, including obviously Edmonton as well. And here you can see that in addition to the raw values of SARS-CoV-2 RNA per mil of wastewater, we can also normalize this relative to the amount of fecal matter in the wastewater by measuring the pepper-mild monovirus, virus that's present in the food supply of humans and only found in human wastewater. We monitor across the province from the very north to the very south. And what we've been able to see is massive regional variation. For example, in our large cities, Calgary and Edmonton, you can see we've had massive rises in the fifth wave of SARS-CoV-2 RNA that have since started to decline. But this isn't universal. If you look at some parts of our province, wastewater values still are on the rise, such as Fort Mac and Medicine Hat as well, and even in Red Deer, which exists between the two large cities. I'll go on to the presentation. We correlate the wastewater values that we measure in real time with clinical cases that are reported to the single service provider across Alberta, Alberta Health Services. And we've been able to look at our wastewater SARS-CoV-2 levels in individual communities. And this is an example looking at Calgary, on samples collected on a day there versus clinical cases that were collected on that day as daily cases or 3, 5, or 7-day rolling averages, and looked at how they correlated with clinical cases after the sample was collected, 1, 2, 3, or 4 days, or days preceding. And our own data locally, as well as that from across the world, generally suggests that wastewater has a 6-day leading indicator to cases. So what we see in wastewater suggests case burden that's going to come in the community 6 days later. And indeed, there are very strong correlations between levels recorded and clinical cases identified. Using this 6-day value, we've been able to develop an algorithm by which to quantify, relative to all cases that would normally be clinically diagnosed, how many we might predict in wastewater. Of course, all cases that are clinically diagnosed do not represent all cases in a community. And here we plotted that over time, where we record the rolling average of 5-day clinical diagnoses versus the wastewater values that were collected and would suggest clinical cases occurring 6 days later, where we show the upper and lower 95% confidence interval boundary of those, showing indeed a very strong overlap, and plotted this at the same time against the test positivity rate. I'll now turn over the presentation to Dr. Pang to take you through more details of our data.
Okay, so thanks, Dr. Parkins, for providing comprehensive background for Alberta Wastewater Surveillance Program. So in my talk, I just want to highlight a few findings in the 2 projects. Next slide. Firstly, I want to talk about the correlation between the level of the SARS-CoV-2 RNA in wastewater and the COVID-19 clinical report cases in different communities in Alberta. So participant wastewater treatment plan facility for Alberta wastewater surveillance has been started in May 2020. And in my talk, 3 wastewater from Calgary was excluded in my talk, since Dr. Parkins already talked about in his presentation. This map shows the 9 wastewater treatment plant location across in province. We are doing the 500 milliliter of the post-grade raw influent and composite wastewater were collected 2 to 3 times per week and shipping on ice to my lab for process, as described by Dr. Parkins. So next slide. What we are analyzed, the 2 data set was generated. Number 1 is SARS-CoV-2 RNA level from 1,762 wastewater sample collected, and the clinical report new cases of the SARS-CoV for the COVID-19 from a local geographic area matched the population served by wastewater from May 2020 to October 2021. The COVID-19 new cases rate per 100,000 population in each sewage shed of the facility were used to create the temporal trend against the level of the SARS-CoV-2 RNA detected in the wastewater. A 7-day rolling average of the new cases in the wastewater of the SARS-CoV-2 RNA was used for investigation of the correction. So the Pearson correlated the co-infants R was used to measure linear correlation between the COVID-19 clinical new cases versus SARS-CoV-2 RNA level in the wastewater in the same population served in that individual wastewater treatment plant. The scale indicated in here are perfect, strong, moderate, weak, and no correlation. Next slide. So the temporal trend of the COVID-19 cases and the SARS-CoV-2 RNA detected in the wastewater showing this figure. Data was combined from 9 wastewater results and match population in clinic cases together to reflect viral infection prevalence of the province community. Blue shadow is clinical new cases, and the orange line is viral RNA level in wastewater. Top indicate the clinical sample testing policy change over time according the pandemic waves. You can see, except the wave 1, the trend of the wastewater results correlated very well to clinical new cases. In the wave 3 and wave 4, the wastewater trend clearly show detect ahead of the time to clinical report cases. Of course, the more detailed the lag analysis ongoing. Next slide. This table showed the correlation between the wastewater results versus clinical cases in 9 wastewater treatment plan corresponding to pandemic 4 waves. Overall, the correlation R was achieved 64%, indicating strong correlation. However, correlation was variable in different communities. There was almost no correlation except 2 wastewater treatment plant facility in wave 1, when that time overall prevalence was very lower. Correlation was increased subsequently since wave 2, and almost perfect correlation was observed in the wave 3, and also the wave 4 even slightly declined. Next slide. These figures with all the data together use a different way to show the correlation. You can see the correlation was perfect in the wave 3, like the bottom, like left corner one. Next one. Next slide, please. So summary, overall, very strong correlation between SARS-CoV-2 RNA level in wastewater and clinical new cases in the community. So was observed, especially during the wave 3. So the impact, I think a factor that impact correlation may include, I think it's number 1, intrinsic difference in the wastewater of each community. Then second one is clinical testing policy, which is changed over time during the study. I think this is major driving our correlations changing. So of course, emerging variants also play very, very important role. As normal, new variants emerging could allow for the wide awareness that allow the disease maker to make the new measures and change the test policy. Another possible reason is new variants may provide different biological characteristics, such as shedding amount and shedding times different. Of course, increased population immunity is another important factor. Of course, include the natural infection as well as a vaccination. For example, vaccination could change the disease burden, result in more asymptomatic infection that is severe shedding the virus in the sewage system, but may be ignored by clinical due to not testing them. So next slide. I just very quickly show 3 slides highlight for VOC detection in the wastewater. Multiplex RT-PCR was used to identify the level of the Delta and Omicron variants in the wastewater sample collected from 11 wastewater treatment plants across the whole province from end of November 2020 to January 20, 2022. Next slide. So this slide, you can see the VOC data shown in this slide was selected from 4 wastewater treatment plants, include Calgary, Edmonton, Red Deer, and Banff, to correlated with the clinical new cases that match to population served in the wastewater treatment plant. The shadow is percentage of the VOC data in blue versus Omicron in orange. The line is temporal clinical new cases in 7-day rolling. you can see the data was 100% in the beginning of in this study period. However, Omicron increased and became predominant quickly after it emerged. Next slide. Summary. Wastewater provides unique opportunity to study the emergency of the VOC since more than one variant could detect in a single sample. So both Delta and Omicron was detected in the most of the wastewater sample after the emergence of Omicron in Alberta. So temporal switch from Delta to Omicron was associated with sharp increase of the COVID-19 cases in the community. So also dates of the emergence of the Omicron variable in the different community. However, there is a similar pattern of the sharp increase in clinical cases after greater than 10% Omicron is detected in the wastewater. So another thing is important. We identify the detection of Omicron wastewater precede the risk in clinical cases. And why the Omicron become like a predominant in all of the community rapidly, however, that are still present in some community at relatively lower level. For example, in Red Deer, they are slowly the Omicron emerging and slowly increase, and data still play very important role. Next slide. We have very big, team. The key people in this program show with the photo. I think this is a nice photo. And however, we would like to acknowledge all the name listed in this slide, even not listed in here, include the research team members, scientific committee members, technical members, collaborating members for their contribution. Of course, we would like to thank all of the institutions and funding agency for financial support and in-kind of support. I think without their support, we cannot stand here to share our study. I stop here. Happy to take some questions. Thanks.
Thank you, Dr. Parkins and Dr. Pang. I think Dr. Bonita Lee will also join for the Q&A session. So we'll just give her co-host ability, and she'll be able to participate. So why don't we start with, why don't we wait until actually Dr. Bonita Lee gets on, and then we can get started, because I know she started to answer some questions, so I'll get her to answer them live. But maybe Dr. Pang and Dr. Parkins, you can maybe provide some of your answers to the first question here is wastewater equally sensitive to all COVID variants?
So, yeah, good. This is a great question. So when we look at our wastewater, of course, from technical point of view, our wastewater, you see that are largely diluted in somehow. We need some minimal infection and people shedding some kind of amount, RNA in the wastewater. Definitely there is some gap. However, when we look at our wastewater, primary PCR results, our VOC sensitivity around 5 to 10 times lower than our primary, the wastewater assay. That is basically I can provide. However, we never directly compare our clinical VOC assay versus our wastewater because we are using different assay to look at. Hopefully answer your question.
Thanks so much. The next one is, are you connecting to similar initiatives in other provinces to share standards, procedures, and data for wastewater monitoring?
So yes, definitely we are. I think both UofC, UofA, we participate in many network wastewater modeling and VOC and routine testing in national and also in Ottawa University and Toronto University. We all those people, engage in a big family. Yes, we are share the data. We are, share, exchange our experience and protocol. Definitely, yes, we do.
And Dr. Parkins, would you like to add anything from?
Yeah, I just comment that it's been incredible the amount of data sharing and technique sharing that goes on. There's weekly meetings that are hosted by the National Microbiology Lab. The Canadian Water Networks had great input into things. There's just been a tremendous experience such that science is shared in real time.
Yeah, it's very, very interesting and great. Are there populations within Alberta that are not being captured, through the wastewater monitoring program that could affect modeling? For example, the Indigenous peoples that are living on reserves.
So our wastewater monitoring program specifically focuses on large urban centers, the one that we've presented, but we do have pilot programs looking at small communities, First Nations communities, and individual facilities of critical importance, whether they're long-term care facilities, hospitals, schools, campuses, residences. Those are all part of our same wastewater monitoring program. We just don't share that data publicly. Those data are restricted to those partner organizations.
Okay, so the next question here is regarding normalizing to PMMOV. Are there seasonal variations in PMMOV quantity? For example, population is being the same, but PMMOV consumption is different for whatever reason.
Yeah, I think this is a great question because in the beginning, a lot of institute, a lot of lab use the PMMOV as a factor to normalization of our wastewater data. However, ongoingly we notify, so not until now, non-factor can be used perfect to normalize our wastewater data. So right now, at least from my personal opinion, even we are using PMMOV to normalize our individual wastewater served population, I think the variation is huge. So from my point of view, there is a site-specific depend on, because a lot of variation, a lot of uncertainty. Wastewater is so complex, and each of the facility has a different characteristic. So I'm not a really normalizing fan. So I prefer use raw data because at least we are not, I recommend don't use one point to estimate what's going on. You use consistent at least the 3 points, say how is go up, is go down. So that is my comment, my personal opinion.
And Dr. Parkins, would you like to add anything to that?
No, I think Dr. Pang's answered that quite well. There are multiple different markers out there that people have explored, whether it's human 18S RNA or whether it's a specific Bacteroides fragilis group members. None of them on the surface seem to improve correlations with clinical cases beyond raw SARS-CoV-2 values. At a population level, it's possible that a subgroup of a population, either neighborhoods or facilities, those correlations might be strengthened, but again, the bloom is off the rose with respect to normalization right now.
All right. We have just about a minute, so we'll try to get through more questions here. Is there any data linked to vaccine status?
We do collection the vaccines, status in our, but we didn't really have time to deeply look at how vaccination is impact our data. However, I really look at the wave 4, what I showed the slide. In that time, so most people already have a second and even third dose. In that time, we see the clinical, reported cases relative underestimated compared to our wastewater. Our wastewater detected much higher compared to the previous wave. So this is why we thought that possibly impact by the vaccination, because vaccine prevent the severe people don't get a test, but the asymptomatic data still go to the wastewater. This is kind of, but we haven't deeply analyzed correlated with the because this is gradually, there is dose 1, dose 2, there is different population and age group. Is a lot of things we need to continue to looking for. Yeah.
So we'll try to get one more in. So can you use your data to estimate the actual number of cases in each community rather than reported cases because of limited testing?
I don't think so. How about Michael, you think? Because we are pool sample, right? We never identify how many, and don't forget, if the who is using diaper in the infants and the senior people and the long-term care facilities, some of kind of condition, it's not belong to our data. That is sure.
Yeah, outside of a cross-sectional survey where you randomly sampled 10% of the population and correlated that, there's no way to have a good benchmark to determine the actual number of cases in a community. So correlating to clinically confirmed cases is the best that we can do right now, recognizing that that is an imperfect metric.
Okay, great. Well, we're going to have to end this session here. We're about a couple minutes over, but I do just want to extend my sincere thanks from the CanCOVID team and also from our audience for your great presentation today. I'm sure that all the work that you've done is very impactful and also going to help the other provinces and territories looking to do the same. So really appreciate your time. And also thank you to Bonita Lee for helping to answer the questions as well. We like to do this both live and through the chat box. So at least when the recording goes on YouTube, others can hear the question and answer period as well. So thanks again, Bonita. I just want to say that please join us for the next speaker series on February 8th from 3 to 3:30, where we will hear from Dr. Janice Keefe on family and staff perspectives on implementing visitation programs in nursing homes during COVID-19 restrictions. And we also have another event. It's a CanPath event, which will be on February 24th from 12 p.m. to 1:30 p.m. That's also going to be held in Eastern time. So hope to see you all back at those events. And thank you so much again, Dr. Pang, Dr. Parkins, and Dr. Lee for the session today.
Thanks for having us in here.
Thank you.
We would like to thank our speakers and members of our network for their continued support and participation in Canada's pandemic response. If you are interested in learning more about CanCOVID or joining our Pan-Canadian COVID-19 Research Network, please visit our website at www.cancovid.ca.